Good evening. This is Journal Club on November 13th, 2025. This is Journal Club number 7, and this is the first time that the current sophomores, the class of 2028, will be presenting. So today's paper is going to be from Science Advances. The annotation of GBA1 has been concealed by protein coding pseudogene GBAP1. And so taking over is going to be Maddie Uluski and Rylee Gray.
Hi.
Hey,
Do you guys see our AI pictures? Yes. Okay. So, welcome to the Journal Club number 7. This is going to be the introduction to Gaucher's disease.
so on our first slide, our whole entire article is basically just about the different genes and their pseudotypes. Sorry, pseudogenes. So there's GBA1, which is its own gene, and then its pseudogene, GBAP1. So that's just the differences between those two. So, and they're both like linked to two different diseases, which we're going to go over in the next slide.
So, this is
So,
wait, are you going?
um, I'll do this one. You could do the next one. So basically, the main idea is that mutations in this GBA1 gene that I just covered cause the Gaucher's and Parkinson's. So basically what Gaucher's disease is, it's not a very popular disease. It's not really heard of because it's a certain genetic gene that you like have to have in order for you to have this disease. So it's where your lysosomes, which are basically like the organelles in your cells that expel all the waste in the proteins, it's when they can't do that. So all that like fatty buildup and all the proteins just like store inside of your body because they don't have anywhere to go. So they kind of slip into like your in between your bones and your brain and just anywhere that it can go because it has nowhere to go. So some of the side effects are fatigue, bone pain, seizures, and more. The fatigue just comes from just being from like the way that your body is with all the fattiness that it just stays there. And then the bone pain is because the fats and proteins get in your bones. And then the seizures is because some of that fat gets into your brain and it stops like how your brain normally works and it causes it to be messed up. And then the treatment is enzyme replacement therapy, substrate reduction therapy, or stem cell transplants. Those aren't really covered in the article, so they're not really important. But Parkinson's disease is a lot more common than Gaucher's disease, and it has no cure. It just has treatment to stop it from getting any worse. Parkinson's is like, when you see an old person and like they're kind of shaky, like they try to eat something and it falls off their plate because their hands are shaking. That's kind of like what it is and what it comes from. And then right here on the right side, we have a picture of somebody with Gaucher's disease. You can see that his stomach is very like enlarged because that's just the way that they look because there's so much buildup in their body that they kind of like swell up. And then this is just a diagram of a lysosome.
This is the confusion with the genes. The GBA1 gene and its pseudogene GBAP1 are 96% identical. This makes it hard for computers to tell which one is which in normal sequencing. As a result, scientists may have been measuring the wrong gene for years. So, with a lot of RNA sequencing, as we can see in the picture, they're not finding every little thing that makes it different. So, with something so identical, they're almost twins. It's, I mean, it'll explain it more on the next slide, but
The next slide really breaks it down.
yeah, it's very hard for them to find the differences between. Can you go to the next slide? I'll keep talking.
Yeah. But just for this first, like the photo, it really breaks down each individual step. So like this is for like the short RNA sequencing. It takes the RNA samples and then it shortens them up But the only problem with this is that it leaves gaps in between the samples. So then it converts the RNA fragments into cDNA, which is just like DNAs. And then on 4, it's ligate sequences adapters and amplify, which I don't really know what that is. But and then the 5th step is it performs the sequencing and then it maps out all the sequencing that you can see.
So in the GBA1 gene, it provides the info for the glucocerebrosidase, which is a lysosomal group of enzymes that catalyze their bond cleavages by reaction to water. It was discovered that its cytogene, GBAP1, makes short-read RNA sequencing difficult because it cannot distinguish the difference between them. So long-read RNA sequencing leaves less gaps in between. When we were talking about the sequencing on the last slide, it leaves less gaps between. It covers more RNA, which allows for less mistakes to be made. So by them doing long RNA sequencing, it's more precise and more like it gets everything that the short RNA sequencing didn't get because of the gaps. As you can see in the picture, there's no gaps in the long reads because they're compared to the short reads, like there's gaps every few. Compare in the reference genome, there's they break it up, there's less gaps and less things missing than in the reference genome when they put it in the long reads, there's no gaps.
Yeah, so when there is sequence gaps, you miss a lot more than what you would with the disease gene. So as you can see, the gray line on the bottom of the long red part, that covers everything. So you could see the entire genome. But then for the short term, there's a bunch of like gaps in between that you don't get the information on that. So people like weren't really sure the differences between the pseudogenes, and they weren't getting as much information as they could have. So this is
Can,
just
can I
what they found.
can I add my 2 cents?
Yes.
Yes.
Do you mind? Can you go back to yeah, that one? Um, so I don't, you probably never thought about this because you're so young and beginning your journeys, all of you, into understanding science and understanding biology. But it's a really, really big deal that we are able to actually decipher the code that DNA uses to build proteins. That's a huge deal because then you can go and manipulate it, which is why Watson and Crick won the Nobel Prize. Once they figured out the structure, they could do that. Okay, cool. But if you knew that ahead of time, you might have then wondered, how did we even do any sequencing at all? Like, how do you sequence a genome? And so here's an analogy I thought was really, really good. Imagine that you have a famous a famous book. Okay, let's the book that they used was, well, I won't tell you. But you have a famous book, and you have a long read of like the book is cut up into pieces, either big chunks of pieces or little tiny, like 3 words at a time. What do you think is going to be easier to assemble? Let me pick a famous book so that you understand.
Catcher in the Rye.
Because nobody would know it, because it's a terrible book. Okay, so this was actually the book that they used when I went to Cold Spring Harbor, which is where Watson was the president. He was in charge. So if you get a bunch of words, and the words are something like this: worst of times, darkness it was, and then you get England, there were fishes, that things, spiritual revelations, it was the, like, how are you going to reassemble that? That's a short read, very hard, right?
Yeah.
But if you get a longer read and you're able to get this: best of times, it was the worst of times, it was the age of wisdom, it was, like, just that much tells me, I'm like, ah, It's The Tale of Two Cities by Dickens, right? Like it's a longer read, it's easier to look at. And so the quickest way we could do it in the past was just these little short pieces. And it's very reliable, okay? Long reads are way harder to do. So because and you really the book is a good example of that. Do you all understand? Cadence.
So, like, do you think that we could have misread like a lot of genes and like we just haven't realized it yet?
We're getting to
Uh,
that, yeah.
yeah, I kind of think, Maddie, that maybe now is, unless you're dead set on it.
Oh, what we talked about before.
Yeah, yeah,
Okay, so basically, we were
on
talking
so
about
many
this.
levels.
We we were talking about this before, but what the scientists that published this paper went out to do is not actually find um like mutation. Can you go back a few, Riley?
Yeah.
Can you go back to the is not to like one more to the yeah, is not to find the mutations in the GBA1 gene that causes Gaucher's and Parkinson's disease, but it was actually to find the differences between genes in their
The
cytogene,
pseudo, yeah.
yeah, the pseudogenes. They were just trying to decipher like what makes them different, but they ended up and they were using this gene, which is a very well studied gene, as an example, and they just ended up finding that the mutation, and they were very successful with what their project was, or their is it an experiment? Yeah,
Yes, it's a series of experiments. Yeah.
So
yeah.
in
Right.
the
So
discussion, like, so wait, sorry, what?
no, go ahead. You finish.
So in the discussion, they really just bring up that the finding of Gaucher's and Parkinson's, it wasn't the main idea, like, at all. Like, that was just something that brought their kind of like experiment to life about figuring out the different pseudogenes. So their main thing was like differentiating pseudogenes versus their normal genes. So yeah.
And they
So
just
then,
ended up finding a major discovery in a very popular disease that affects a lot of people.
mhm. And so circling back to Cadence's point, Cadence, that is dead on. That's the point. Is wait a minute. Are you saying that we may have misread everything so far? Yes. So that is a huge deal. This paper, the premise of this paper is better than the one I did 2 weeks ago. So, all right, go ahead.
Okay, can we go? Yeah.
Okay, so maybe I'll do what they found and then you could do why it matters.
Okay.
So they found over 30 new versions of the GBA1 gene because of um this long read RNA sequencing. They found 30 new versions and over 40 new versions of the GBAP1, the pseudo gene. So they found 30 new versions of the normal gene and then 40 of the pseudo gene. And then some of these genes can make proteins, even though before they thought that the pseudo gene was non-functional. And they also found that these new proteins don't break down fats, but they might do something else. So we just don't know what yet.
And why it matters is because the mistakes in gene mapping can affect disease research like Parkinson's or Gaucher's, as well as genetic testing, which a lot of people get done, and drug development. Because, like, understanding DNA sequencing and genes and RNA sequencing can help with drug development and finding better medicines and treatments and cures for disease research and like just sicknesses. And some mutations might behave differently because they're in these new transcripts.
So I feel like in order for us to understand them better, we just need to do, not like us, but like scientists in general, they just need more research on it to like fully understand what all of these genes are capable of having. So now like the big picture is, and what everything that we've been talking about is that many other genes may have hidden pseudogenes that are confusing scientists, which is basically what Cadence said when she brought up her question. So that's just the big thing. And then fixing these mistakes with long-read sequencing can help us understand how genes actually are. It can improve diagnosis and help RNA-based treatments. So it's just overall like RNA long-read sequencing just can overall like expand our knowledge on genes.
Yeah.
So me and Maddie are gonna present figure one, and then I'll be asking who wants to do all the other figures. Okay, let me just go and get them. Okay, so in figure 1A, which is this one to the left, what I got from it is the top that has the gray, yellow, and purple genes. That's all GBA1. And um, this example is basically just why short read DNA doesn't work. Because down here at the bottom of the gene, the mRNA, that's the GBAP1 gene, the pseudogene, sorry. So as you can see, the purple and the gray, or the white, gray, I don't know what that is, they're extremely similar, which makes it very hard to see the difference between them. So this is just really why short read doesn't work, because it's too simple and it doesn't break down the genes as much as it should. Yeah.
What is a
Mm,
pseudogene?
It's like a mutation that happens over time to a
We
certain
have
gene.
the definition in here. You want to go back a few? It's right there. Go back. One more. It's the same DNA sequence as the gene, but with mutations.
Okay. And how does that happen?
It's just something that happens over time.
Natural selection.
Kira has her hand up.
Yes, Kira.
A pseudogene is a gene that doesn't function.
Usually, yeah.
Okay,
I mean,
so like,
you know how it is part of evolution. Yes, it happens. But if you go forward a slide or two, one more, go to the figure one there. Do you notice on the top it says that as a gray arrow says duplication? Because
yeah,
what happens is sometimes the proteins that are supposed to be replicating the DNA, if the DNA twists on itself, they then don't realize that and they jump onto it again and make a copy again. So they're right next to each other. But the thing is that that second one is not useful and could even be detrimental. If you have too much of something, that's bad. You need 100% of what you're supposed to have. If you have more than 100%, you get problems. If you have less than 100%, you get problems. Think about like Down syndrome, right? Trisomy 21 is bad because you have 150% of all of the proteins, the gene products on chromosome 21, 150% because you have 3 chromosomes. You're supposed to have 2. And so that's why this happens. But evolution also then selects for like advantageous changes. So if you think about things that are interesting to you, dopamine transporter, for example, you all know what dopamine is. The dopamine transporter is actually evolutionarily related, is a duplication of the serotonin transporter, but doesn't work with serotonin. It works with dopamine more. So that's how that happens. A pseudogene is one that doesn't quite function. So Maddie and Rylee, especially, do you see, and maybe Cadence then, right? Do you see how that's like, it's not supposed to function? It's not supposed to even make protein. Do you see? Like if we go to the next figure 3, something like that. Anyway, I'm setting it up. Go ahead.
So like it's so crazy that it does work because it's not supposed to.
Right.
Okay.
You're not supposed to get any protein.
Yeah.
It's supposed to be essentially just a ghost gene that was copied and just sits there.
Has Cadence.
Because since he brought up Down syndrome, is like, does this like affect people with Down syndrome worse than it affects people like with the normal amount of like chromosomes and DNA?
Well, that's a good question.
If you know. I'm not trying to like make it seem like you don't know what you're talking about. I'm just like curious.
No. I need a piece of information before I can answer that. What chromosome, and I didn't see this in the paper, so someone's probably gonna have to Google it. What chromosome is GBA1 on? GBA1 and GBAP1 are on the same chromosome right next to each other. Which chromosome is it?
Oh,
Q 21.
Is it really?
Or one Q21, sorry. Yeah.
Did you know that, kids?
No, I don't know what that is.
isn't that the Down
It's the
syndrome
20.
gene?
Yes, it's the Down syndrome. Wow. Okay. I was just using
Crazy.
Down syndrome because it's the most well known of the accidental duplications. But if this is on the 21st chromosome, then yes, Cadence, it would definitely be a candidate for problems with people who have Down syndrome. Yes.
So it'd be worse
Um,
for people with Down syndrome.
Well, if you look up classical, Down syndrome was working on Down syndrome was my first postdoc, and it has classical symptoms. People with Down syndrome are shorter in stature, which is not what this paper is about. But think about this: they have this weird crease in their hand. Their tongues are bigger in their mouth than most people. They have the classic Down syndrome facial features that you would recognize, and almost 100. So their life expectancy is in the mid-50s, and almost 100% of them get early onset Alzheimer's.
Because it's on the same chromosome?
That's what led us to believe that the Alzheimer, if there's a genetic component to Alzheimer's, that it must be on or related to the 21st chromosome. But they also have motor disorders, and if you actually see how Gaucher's happens, it's quite possible that what they're calling Alzheimer's isn't, and instead it's Parkinson's, and that we're misdiagnosing it.
And it could be just a different not like mutation, but a different type of Parkinson's that we just haven't fully.
Well, one of the main symptoms of Parkinson's is dementia that looks
Yeah.
like Alzheimer's, right?
Yeah,
Does that interest you, Cadence?
but
Because if
it's
it does,
also motor, right? Because we know
it doesn't
that,
have to be. It doesn't have
but
to
we
be.
know that people with Down syndrome have motor.
They can,
They
but
they
the
they
classic symptom is dementia, not the motor. They can get the motor, but it's dementia.
but,
Almost 100% of them
but
get
motor is
some form of
also a
dementia.
symptom of Parkinson's.
It is.
So maybe the motor doesn't show up for everyone.
It actually usually shows up after dementia in most people who have Parkinson's. So the reason I'm asking Cadence is if this interests you, because you seem to be kind of nailing this, put it in your back pocket as a subject to do your capstone on. Okay. All right. Sorry, I keep interrupting, but that's interesting. That's good. Go. I'll shut up.
Okay, so figure 1.D, it's 4 on there, but it's 1.D. So the protein 3D structure predictions, that's just like the predictions of what we think the gene will look like and what its structure will be. And then the translation in, I don't know how to say that, but colocalization.
Co-local, an area, co-localization.
Colocalization.
Yeah.
Okay.
Where are they going to be produced? And they were mostly looking at the brain.
Yeah.
And you should take credit for this, the two of you. Did you choose this paper because it was about protein folding and you knew that the honors students were doing protein folding this past couple weeks?
No.
Yeah.
Yeah,
Oh.
No,
you
man, we
totally
had to say
did.
yes.
Oh, yes.
Yeah. So freshmen, Do you see the alpha helices? Do you see how it's folded?
Yeah.
Right? A slight, and you see how there's a slight difference between GBA1 and GPAP1. Rylee, can you show the loop on the right of that? Can you like point it out with your mouse? Yeah. See how that's missing? Right? Or it actually has it in GPAP, but not in the real gene. Sequence equals structure equals function. You change it just a little bit, and all of a sudden, the protein's not supposed to work, supposedly.
But it does.
But it might. Yeah. Go ahead. Oh, can I say that the cell type thing was one of the cooler things in this paper? I don't think
I didn't
anyone else
really
would
understand the
care
cell
about that.
type selectivity. I thought it was
Yeah.
just like determining what the cell would be made up of right there. I
No.
wasn't really sure about that one.
So what they did was I'm gonna 2 summers from now, I'm actually gonna teach you how to do part of this. This is called it's called FACS, fluorescent assisted cell sorting. So what you do is you have a tissue like a brain, and you have antibodies for these two proteins, and then you attach some sort of fluorescent probe to that antibody, so that wherever it glows in the dark. So those of you who are doing chemistry right now for Mr. Cream should know what fluorescence is, because you shoot a photon at something and it excites the electrons, right? This should sound familiar, chemistry kids, right? And then as they relax back down, the energy has to go somewhere, so it shoots off another photon of a different color. So if you're in a black light room, for example, and you're wearing white socks, your socks don't show up white in the black light. Instead, the UV light from the black light um, increases the energy in the socks, really the detergent in the socks, and then it releases a purple color. So that's why your socks glow under a black light, fluorescence. And so you would use this thing to attach to whoever is producing the protein, and then you sort them by type. And you can see that they actually are being expressed in, and it was really hard to see because it's really grainy. It was really, really grainy. Like, this is a bad figure, just like they did last time. Science advances, for some reason, they don't have good figures. Microglia, which are cells that do cleanup in the brain, neurons, interneurons, astrocytes, and oligodendrocytes. So those are all actually all of the brain cell types, and apparently they all produce these proteins. Next.
Okay, does anybody want to do figure two? Oh, okay, here, you could do figure two.
Okay, figure 2 is the least important figure, and it's also like so the thing about this figure is it's an online analysis, meaning they didn't do any of these experiments. Like, this is not really their own work. Um, this is just taken from online sources. And then a couple of things you might want to know when you're reading like these graphs and stuff is a pseudogene is a gene that generally does not function properly. And an expression is when a gene turns on, and on means it's made a protein. There are 21,000 genes, but there are only 100,000 proteins, and you can get that amount of proteins from alternative splicing. And alternative splicing is the process of when a single gene can make multiple proteins. And like an analogy for that would be like if you're making bread from a recipe, you can like alter some of the ingredients and come up with like an alternative kind of bread. Okay, so some of the results are for figure 2A, it shows what's been processed versus unprocessed in the human pseudogenes. Does that make sense?
Do you know what does like processed and unprocessed mean? Does it like what's being Char, do you know what's being processed?
Are they being spliced or not? So, are they alternatively spliced? So I haven't gotten here with the freshmen, but you sophomores and juniors should remember that. Well, Kira, didn't you just say, right? There's 100,000-ish
Yeah.
genes, or there's 100,000-ish proteins, but there's only 21,000 recipes. Like, If it's a 1-to-1, why are there 5 times as many proteins? It's
Because of
because you
alternative.
actually, yeah, you just take you take the recipe, but you like change an ingredient. So you imagine if you're going with the analogy that Kira was using about bread, you have regular bread, right? But let's say, what if you remove the yeast from it? What do you get? Well, you get flatbread,
Jewish
right?
bread.
Jewish unleavened matzah. What if you add extra yeast and maybe a little sugar? You get like sweet rolls. Like it's bread, but you've just slightly changed the recipe, and it's in the recipe. But you're there are certain proteins that are choosing whether you're going to use all. Let's say that the thing is 10 exons long. You're using, so sometimes you'll use all 10. Sometimes you'll use exons 1, 3, 5, 6, 7, 8, 9, 10. You'll
Six,
skip
seven.
some, right? Alternatively, that's what it is. Is the protein processed, or is the RNA processed or not? That's all it's looking at.
Rylee, can I go back to the figure? I didn't finish.
Yeah, sorry.
So the graph for B is of the parent genes, and it shows the percentage on genes in the data that are known to cause diseases. So those are only ones that are known to be causing diseases. It doesn't mean there's not more. For 2C, that graph shows the gene expression and how it's ubiquitous, meaning found everywhere.
Ubiquitous.
Did I say it wrong?
Yeah, you were close enough.
Ubiquitous?
Yeah, it's ubiquitous. Yeah.
Okay. And then 2D is the percentage of pseudogenes that are alternatively spliced, showing the proportion of the pseudogenes that are functionally important.
In different
Thanks, Kira.
types, right? In
Yeah.
different organs. Yeah. So like the top one's brain, where it seems to be really important. But it seems similar in the other ones too. Heart, lung. Okay. Thanks, Kara.
Yeah, I chose the easiest one.
Thanks, Kira. Okay, does anybody want to do figure 3?
I'll go.
Okay.
All right, so if you look at figure 3A, it is a histogram, aka bar graph, that shows, like the pseudogene and gene pairs between like everything. So if you look at the graph, most pseudogene genes have like an 80, somewhere around like 80%. That's for the or 90%. That's for the graph peaks. That's mostly common for pseudogene to see. However, GBA1 and GBAP1, they actually have a 96% similarity, which is way too high for those two. So since these two are so similar, when you use different types of sequencing, aka the shorter read sequencing or the one that's inaccurate, you get they become confused and it creates a big mess. Now for freebie, it has two plots, which are frontal cortex and hippocampus, which is inside your brain. So the main thing about this graph is that when you see the gene expression, which is basically how genes are like how they're utilized to make a protein, it doesn't look big because if you look for the frontal cortex, for example, the peak of GBAP1 seems to be much shorter than GBA1. But if you actually compare these two and you do the math out, you get something like about 1.2 to 1.3 times difference, which is also, once again, very small, but for the relationship between these two, because GB1 is the more important gene that should be used. But again, GBAP is pseudogene and should not be expressed like very closely to it too. So it's basically saying that the short read sequencing says that GBA1 and GBAP have really close expression levels. That is not good and it is wrong. And then for the 3C over here we have a little we also have another plot, which is a graph of a log2 thing. I'm pretty sure it's a logarithmic function, doesn't really matter right now, but the black line means how the shorter read sequencing read the pseudogene, and the red line is how the Oxford nanopore long read sequencing technology measure this, which is a more correct and accurate version. So if you look at the graph right here, you can see that the difference between the two on the black line is actually very, very, very similar. 0.001 in the hippocampus and 0.003 in the frontal cortex. The 1, 2, and 3, the positive values mean that the GBA1 has a higher expression than GBAP1, and then 0 is like the more zero equal expression, and then negative means that GBAP1 seems higher. But since the black dotted line is really close to 0, that must mean they don't have it. They have a little similarity and they're equally expressed. Now for the red dotted line, which is more accurate and correct, it is much, much, much higher and is more tailored towards GBA1, which is what we want. And that must mean GBA1 is actually much higher than GBAP1. So short read sequencing, it will underestimate GBA1, and it will also equally take GBAP1 and say that they're pretty similar and that they should be used the same way.
Thanks, Andrew. So basically what you're saying is that like the short read sequencing shows less information than the long term, or the long read sequencing.
Yeah, A shows the similarity, then B shows the like inaccurate measurements because of that similarity, and then C shows that the long read sequencing and the whatever ONT technology is much, much better and accurate than the older short read sequencing technology that we used.
Thank you.
Yeah, thank you.
Crap. Good job. Jeez. We were at lunch, we were trying to figure out what the heck B was. I was like, I didn't even know how to read that graph. But for those of you who did like sophomores, you've actually done something like this already. When you do your lab, pretend that so this is called a violin graph. Pretend that the violin shapes are not there. What do you see? Just box and whiskers, right?
Mm-hmm.
That's it. It's a box and whiskers plot. So the violin gives you extra information about the data set, but what matters is the box and whiskers, and that you see up above it says Wilcoxon and the p-value, which you should remember how to read. And freshmen, you'll learn how to read that in about a month. Good job, Andrew.
Thank you, Doctor.
Okay, so does anybody want to do figure 4? All right, Chris, you go ahead.
All right, so figure A is counting the GBA1 transcript, things that were like identified in each transcript category. And the blue is the coding known, which is protein coding GBA1 transcripts. It's telling us that protein coding GBA1 transcripts exist. And then the green, which is the coding novel, has a large number of newly previously unannotated GBA1 coding transcripts and it tells us. Hold on, sorry. It tells us that there's a large number of newly previously unannotated GBA1 transcripts found, and it also is also the largest category for GBA1. And the red is the NMD novel, and these novel transcripts are predicted to undergo nonsense meditated decay. And then the purple and the light purple are uncoded, known and uncoding novel, and they both have small amounts. And then for figure D, it's similar to the first one, but it's GBAP1 and pseudogenes instead of the PBA1. Oh, sorry, PBAP1 pseudogenes instead of the PBA1. And the blue is coding known and is like very few, which is expected because it's the pseudogene. And the green is the coding novel, which is also none, which is also expected. The red is NMD novel, and it has a large amount, and most are novel. and expected to undergo NMD. And the purple and light purple are uncoding known, which is very few. And then the light purple is uncoding novel and has a medium number of newly discovered uncoding transcripts.
Okay, thank you.
Thank you.
So now on to figure 5. Does anybody want to do figure 5?
In like a sentence though, Rylee, like what's the take home from this?
From the 4th one?
Yeah, in a sentence.
So, sorry, wait, what'd you say?
In like one sentence, why have a figure showing this? Like what was the point?
So what I see, I think that there's like less uniqueness in the pseudogene from what G shows. Because there's higher, like, no unique transcripts. I don't really, what does it mean by transcripts? Like, is that just like what the genes are made up of?
mRNA. No, mRNA. So when you go and you express a gene, the expressed thing is RNA. It's mRNA that leaves the nucleus, and that goes on to tell the ribosomes, here's how to make the protein. So you can measure those transcripts. But in the interest of time, let me just say, they did not expect to find coding anything in the pseudogene. Right?
Yeah.
It's not supposed to code for anything. So this was a surprise. Okay. Sorry.
Because it codes, does it mean that it works, like it does stuff and it is functional?
What is the it you're talking about?
The pseudogene.
Now you have to kind of speculate that it's possible.
Okay.
Yeah.
Yeah.
And they didn't expect. So going back to Cadence's comment, which was right on, it's if you have what is it now? 60 something years of thinking it's this one gene that's contributing to it, and now all of a sudden you have to go, wait a second, it might also be this other gene and we just didn't notice because they have the same sequence. Right? So it would go far to explain a lot of stuff that was sort of weird about Gauchers.
Okay, so does anybody, okay, Bill, do you want to do it?
Yeah. Okay. So for figure 5, letter A, the top one there, it's kind of hard to see on the slide, but the top one there,
Sorry, it's kind of blurry.
yeah, it's they called it the main one, MANE. But that was just like the most common type that they found. And then under that is just different transcripts, which is just different ways the gene is expressed. And those are like the 3 common ones. You can see that they're like everything in red there is something that it's different from the main one. And then in the blue is something that it has that the main didn't have. Yeah, and then B does the same thing with GBAP1. Again, they call this one the canonical one, which is just like the most common one again. And again, you can see for the other 2, they're either missing things or they have like are including things that aren't in the other one. So I was kind of thinking that maybe like missing something like in their genome would be like a cause for like something like Gaucher's disease or something like that, that they're missing like the proteins they need to be making. But then we have C, which is kind of like the main one. It's kind of like in the title, but it says, so you have like the C and it says glycohydro 30C, and then you have the big green one, which is glycohydro 30. The glycohydro 30 is the domain, which is different among the GBA1 and GBAP1. So they have like everything else is similar except for the domain. And that's why they're having like different functions, and they look the same, but they still have, like different functions. Then D, I was a little confused because it looks a lot like E, but they called it the X-ray of it. But I was a little confused why they had both D and E. But then E is kind of just like visual representation. E and F is a visual representation of A. So E is the top one in A, and then F is just the three following. You can kind of just see
I
that
know the
there's
answer. When
okay.
it says X-ray, that's actually been experimentally verified using
Okay.
X-ray crystallography, like Rosalind Franklin used to do. So D is the only one that we actually know the structure of. All the
Yeah,
rest of
and
them,
G, right?
yes, sorry, E and
Okay.
G, yeah,
Yeah.
but sorry, D and G, E, F, H, those are all they're all computer software trying to predict. It's a prediction from
Okay.
computer software. Yeah.
Okay. So yeah. So E and F just visual representation for A, and then same thing for G and H. So G is the top one, H is the next two, and there's also a little like I don't know if you called that like a legend or something, but the color is just basically how like confident they are. So for E and F, they're pretty confident for that. Uh, and then H, it kind of varies, but less confident than E and F.
So
That's about it.
do you think I don't know this, but do you think the reason that like E, F, and then the two that come after that, do you think the reason why they kind of get like less, I don't know what like the word for it, but like there's less
Just
going
like their
on?
buildup is less. Like
Yeah,
there's
because of
less.
what it shows, they lose things every single time. That's why
Yeah.
they're not.
When you're saying losing things, what
They
you
just
mean,
don't, I
what
don't
you
know
mean
what
is
they're
they're
called, like the,
alternatively splicing it. Again, if you have exons, it's you've got one recipe, exon, and an exon is the coding part, right? So lines of a recipe where it's how to make bread. Step one, get the flour. Step two, get the water, right? You have all these steps. Except in alternative splicing, you actually skip some of the steps. So if you skip, for example, in F, the one on the right, you have skipped steps 5 through 10. So you haven't made the whole thing. You've only made a little chunk of it. Do you see?
so each
And
of them are
so,
just like little chunks of
yes,
E.
and functional pieces of protein, functional pieces are called domains. And when those domains have a certain shape to them that actually does something, that's called a motif, which anyone who's a musician should know what a motif is. Something that sort of comes up every once in a while in a musical piece, something that comes up every once in a while in a protein that does a certain thing. If we see that motif, then we know it does this thing. That's why this is my favorite figure. I think this is sequence equals structure equals function. This is a really great example of that.
So how would you explain like what's in blue in A and B, the ones that are in like the bottom three but aren't in like the main one?
Ones that are in the bottom three, but not
So
in
like
the
of
main.
A, you know what I mean?
Of A.
Yeah, I mean like panel A or
Yeah.
5A.
In early experiments that we used to do, we would we would actually remove and kind of pretending to do alternative splicing, which I don't even know if I still don't think we know how to control it, but you can actually just say, I'm just going to get rid of this part of the gene and see what it does. And that's kind of how I'm reading this. Is like you have sometimes just a piece of the protein. And the assumption was because it's a pseudogene for GBAP, it's not going to do anything. Again, maybe that's the wrong assumption, you know? But I'm not an expert on crystallography, as I always say whenever we talk about Rosalind Franklin, so I'm not really sure. You got two hands up.
Wait, can you guys still see my screen?
Yeah.
Okay. Yeah, Andrew.
Just for 5D when it's talking about like the yellow part, it's talking about glutamate because I noticed it said glue residue. I don't know if it's talking about just like glutamate specifically or something like that.
You haven't been able to hear me at all, have you?
Nope.
Why didn't you say anything?
I thought
'Cause
my
I was
volume was
happy.
working.
I thought I was
It
going
didn't even tell me we were
Same.
talking.
I'm here having a panic attack because I'm going freshman. You just had a test today on this. What is a protein made of? No one's responding. I'm like, you just had a test today. Of course. What is it?
Amino acids.
Amino acids. Glutamate's an amino acid. That's all.
Okay.
It's just referring to the amino acid. That's all. And here I am panicking that none of you know because none of you're all embarrassed to answer, but you actually can't hear me.
I was so
Next time, just be like, Char, we can't hear you.
I thought I was going crazy. Like I was turning my camera off and on.
Me too.
Yeah, I'm using a button to control mine because it's faster and easier for me. Cadence.
I was just curious on what the difference between like a domain and an enzyme is, because they sound similar. Like based on your definition of them being
Yeah.
like a protein that does stuff.
An enzyme is a full protein that does stuff. A domain is a part of the enzyme. It's a part. It's the part of the enzyme that actually is probably the most important to do the stuff. Like if I said, somebody's, I'm hearing an echo of myself. If I said a human is really good at hitting a baseball or can hit a baseball, right? So a human does stuff, and a domain would be your hands or arms, right? If I cut off your hands or arms, you're still a human, but you're not able to do the thing anymore. So what is it? Which part of the protein does the stuff? It's the motif, or it's the domain that contains certain motifs that do it. Just different sizes within the protein. Okay. We're running out of time here. So what were you doing next, guys?
We're overtime.
Yeah.
Yeah. Did
Have-
Rylee,
You what?
we have the conclusion?
Yeah.
Yeah.
I'll
So
go.
the conclusion's kind of simple.
This discovery helped realize that mutations in the GBA1 gene can be hard to find, but can have significant effects on genetic diseases such as Gaucher's and Parkinson's. Our new favorite word is pseudogene. And does this paper belong in Science Daily? Yes, because the journal impact factor was a 12.5.
What's Science
That's
Daily?
from Chet. What?
It's Science Advances. You meant Science Advances.
Oh,
Okay.
And didn't we look it up the other day and it was a 4 point something? It wasn't 12, is it?
I
No,
thought
I
it was,
think that was a different article.
yeah, this is a 12. This is a,
Oh, then that makes more sense. Okay, good. All right. So yeah, I completely agree. I think this might actually be good enough to be in a better journal. I liked it.
Yeah, this one like was not boring.
Yeah, this wasn't boring
In the
like
best way
usual.
possible.
Yeah. And I had advised them to skip figure 6, and you really could skip 7 and 8 also because the main points were
You could
made
skip
in the earlier
2.
figures. You could skip 2 also, yeah. The main points are like set up in figure 1, and then data in 3, 4, and 5, and then you're good. It's a pretty good paper. So the next people to go are Dylan and who?
Iris.
Iris. Okay, so you guys should pick a paper, even though it's in January. You should still pick a paper, and that way we can get ahead. So, hey, good job, guys. That was good. That was very good. Yay!
Thank
Okay.
you for everybody who
Um,
volunteered too.
Thank you, everybody.
Yeah, I told you guys this'll get fun once you get the hang of it and you actually own it. And I think even if you don't agree with me that you did that today, please leave me with the illusion that you did because I'm very pleased. So if they can be like this going forward, they will continue to get better. Um, and we'll purposely pick papers that are really good, and we'll purposely pick papers that are really atrocious because part of the fun of it is breaking down a paper and be like, this is garbage, and here's why. Blah, 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 blah. This paper was not garbage. It was really good.
Yeah, like the one with the mice was pretty bad.
Yeah.
Do you remember that?
Yes. Yeah. Anybody have anything else?
Did you guys like this article a little bit? Like, was this at least a little bit interesting?
It was interesting. And you and Maddie might feel differently, but it was nice like that other people did it and not Char. Because I kind of, no offense, but I kind of get lost in your words sometimes. Because like you'll be like, oh, I don't understand this, and then you'll explain it, and I don't understand the words you used when you explained it because your vocabulary is so much more like advanced. So like I get kind of lost, but like this one actually made sense.
Yeah, that's what I was gonna ask. Like, did it make more sense to understand?
Yeah, definitely.
We're going to be using at least one of these techniques 2 summers from now. I'm going to teach you how to do it. So that should be fun. But in the meantime, we have to buy some microscopes and stuff. Like you might think I had mentioned that I spent a lot of money this week on stuff for dark sky, which no one wants to chair, and a lot of money on 3D printers and stuff. The telescopes used in dark sky, if you learn to operate them, first of all, it's fun, but if you learn to operate them, then you will actually know how to operate a microscope. So there is method to my madness. So you get the fun part, and I'm going to ask Dr. Pellegrino if we can maybe build a fire pit in the back. That way we can do stuff.
I thought you wanted to build an observatory.
That too.
I thought you wanted to go.
Kira was always trying to leave. Yeah.
I have
I'm not
work
going
to
anywhere.
do.
Yeah. All right. Well, thank you all again. Great job, Rylee and Maddie. Thank you. I have pictures I'll send you.
Okay.
Good job.
Okay. Thank you guys so much.
See ya.
Guys.
Bye, guys.
Have a good night, everyone.